Deep in the Australian outback, geologists have discovered rocks that contain something impossible. Crystals formed 4.4 billion years ago, barely 100 million years after Earth's birth, showing chemical signatures that indicate liquid water existed on our planet's surface when it should have been a molten hellscape. These ancient zircon crystals tell us that everything we thought we knew about Earth's violent beginning may be wrong. Our planet's story is not what the textbooks claim. Today, we compress 4.6 billion years of planetary evolution into less than 1 hour, revealing catastrophes that reshaped continents, extinction events that eliminated 96% of all life, and evolutionary leaps so dramatic they appear engineered rather than natural. Prepare yourselves. We begin. In the beginning, there was chaos. 4.6 billion years ago, our solar system existed only as a massive cloud of gas and dust spinning through the void of space. This nebula contained the remnants of previous stars that had lived, died, and exploded, scattering heavy elements across the cosmos. Within this cosmic debris field, gravity began its patient work, pulling matter together into increasingly dense concentrations. The proto-sun ignited at the center of this swirling disk, its nuclear fusion flooding the inner solar system with radiation and solar wind. But surrounding this newborn star, countless smaller aggregations of matter were forming. Now, these planetesimals, rocky bodies ranging from pebbles to mountains, orbited the young sun in a crowded, chaotic dance. Collisions were inevitable and constant. Earth began as one of these planetesimals, growing through a process scientists call accretion. Each collision added mass, and as Earth's gravity increased, it attracted larger and larger objects. The planet grew rapidly, achieving most of its current mass within just 10 to 20 million years. This timescale seems vast to human perception, but represents merely 0.2% of Earth's ultimate age. The violence of this formation process cannot be overstated. Planetesimals struck the growing Earth at velocities exceeding 20 kilometers per second, releasing energy equivalent to millions of nuclear weapons with each impact. These collisions generated such tremendous heat that the entire planet existed as an ocean of molten rock. The surface temperature exceeded 2,000 degrees Celsius, hot enough to vaporize iron and turn rock into liquid. But the most dramatic event in Earth's formation was yet to come. Approximately 4.5 billion years ago, a Mars-sized planetesimal that scientists have named Theia collided with the proto-Earth in an impact so catastrophic it nearly destroyed both bodies. Computer simulations of this giant impact reveal the stunning violence of the collision. Theia struck Earth at an oblique angle, liquefying both planets instantly. The energy released by this collision vaporized enormous quantities of rock, creating a massive debris cloud that surrounded Earth. Within hours, this debris began to coalesce under its own gravity, forming a ring system around our planet similar to Saturn's rings, but composed of vaporized rock rather than ice. Within centuries, this ring material aggregated into a single body that we now call the Moon. The Moon formed remarkably close to Earth, perhaps only 15,000 kilometers away, compared to its current distance of 384,000 kilometers. This proximity meant that the Moon appeared 15 times larger in Earth's sky than it does today, dominating the heavens with a presence both beautiful and terrifying. The tidal forces generated by this massive nearby body were equally dramatic, creating ocean tides hundreds of meters high that swept around the planet every few hours. The giant impact had profound consequences for Earth's subsequent evolution. The collision tilted Earth's rotational axis by approximately 23 degrees, giving our planet the axial tilt responsible for seasonal variations that would later prove crucial for the development of complex life. The impact also likely stripped away much of Earth's original atmosphere, forcing the planet to develop a completely new atmospheric system from volcanic outgassing and later cometary impacts. Analysis of lunar samples returned by Apollo astronauts reveals that the Moon formed from material that was thoroughly mixed between proto-Earth and Theia. The oxygen isotope ratios in lunar rocks match Earth's ratios almost perfectly, indicating that both bodies were liquefied and combined during the impact. This discovery confirms that the Moon represents Earth's lost twin, a planetary body that merged with our world in the most violent collision in our planet's history. But here's where the mystery deepens. Those ancient zircon crystals from Australia that opened our story contain oxygen isotope ratios indicating they crystallized in the presence of liquid water. These crystals formed only 100 to 160 million years after the solar system's birth, suggesting that Earth's surface cooled from molten rock to temperatures supporting liquid water in an impossibly short time. Traditional models predicted that Earth should have remained molten for at least 500 million years after the giant impact. The rapid cooling indicated by zircon evidence suggests either that the impact was less energetic than models predict, or that Earth possessed cooling mechanisms we don't yet understand. The planet appears to have been in a tremendous hurry to develop conditions suitable for life. The evidence extends beyond these controversial zircons. Chemical signatures in Earth's oldest rocks suggest that plate tectonics, the process that recycles Earth's crust and regulates atmospheric chemistry, may have begun operating within 200 million years of planetary formation. This timing seems impossibly rapid for a planet that should have been cooling from a molten state. Some scientists have proposed that Earth's early cooling was accelerated by a thick atmosphere of water vapor that acted as a thermal blanket. It was radiating heat into space more efficiently than the vacuum above a bare molten surface. Others suggest that early plate tectonics provided convective cooling that removed heat from the interior far more rapidly than conduction alone could accomplish. Regardless of the mechanisms involved, the evidence indicates that Earth transitioned from a molten hell world to a planet with liquid water oceans in less than 300 million years. This transformation set the stage for the next chapter in planetary evolution, a period of bombardment that would both threaten and enable the emergence of life. The Late Heavy Bombardment. Between 4.1 and 3.8 billion years ago, Earth experienced a catastrophic increase in asteroid and comet impacts that scientists call the Late Heavy Bombardment. Evidence for this bombardment comes primarily from lunar samples that show a dramatic spike in impact melting during this period. Since the Moon preserves impact records that Earth's active geology has erased, lunar craters provide our primary window into this violent epoch. The bombardment was intense beyond comprehension. Computer models suggest that Earth was struck by at least 17,000 asteroids larger than 10 kilometers in diameter during this 300 million year period. Each impact released energy equivalent to 100 million megatons of TNT, enough to vaporize oceans and sterilize the planet's surface. Some impactors exceeded 400 kilometers in diameter, large enough to create craters spanning entire continents. The origin of this bombardment remains controversial. The Nice model, named after the French city where it was developed, proposes that gravitational interactions between Jupiter and Saturn caused those giant planets to migrate to slightly different orbits. This migration disrupted the asteroid belt and scattered icy bodies from the outer solar system, sending a rain of debris toward the inner planets that lasted for hundreds of millions of years. Alternative theories suggest that the bombardment never actually increased in intensity, but merely represents the tail end of planetary formation when large impactors became rare enough to be individually recorded in the geological record. This interpretation suggests that Earth experienced continuous heavy bombardment from its formation until approximately 3.8 billion years ago, when the solar system finally cleared enough debris to allow stable surface conditions. Regardless of its cause, the late heavy bombardment had profound effects on Earth's early evolution. Each major impact vaporized enormous volumes of ocean water, creating steam atmospheres that could persist for thousands of years before condensing back into liquid water. The largest impacts may have completely sterilized Earth's surface, boiling away the oceans entirely and heating the atmosphere to temperatures exceeding 1,000 degrees Celsius. But the bombardment also brought gifts. Many of the impacting objects were comets and carbonaceous asteroids containing water, organic molecules, and volatile elements. These impacts delivered the raw materials necessary for life, including carbon, nitrogen, phosphorus, and complex organic compounds that might have served as precursors for biological chemistry. The timing of the bombardment's end correlates suspiciously well with the appearance of the first evidence for life on Earth. The oldest potential fossils date to approximately 3.8 to 3.5 billion years ago, suggesting that life emerged almost immediately after the bombardment ceased. This correlation indicates either that life required stable surface conditions to develop, or that the bombardment itself repeatedly sterilized Earth, so forcing life to originate multiple times before finally establishing a permanent foothold. Recent discoveries have complicated this picture. Chemical signatures in rocks from Greenland, dating to 3.8 billion years ago, show carbon isotope ratios that some scientists interpret as evidence of biological activity. If this interpretation is correct, then life existed on Earth even during the late heavy bombardment, surviving the impacts by retreating into protected environments like deep ocean hydrothermal vents or subsurface rock formations. The bombardment may have actually accelerated the origin of life by creating extreme environmental gradients and chemical disequilibria that provided energy sources for early metabolic systems. Impact-generated hydrothermal systems could have created ideal chemical reactors where organic molecules concentrated and polymerized into increasingly complex structures. Deep drilling projects have recovered rock samples from impact structures created during this period that contain unusual organic compounds and chemical signatures, suggesting hydrothermal activity persisted for thousands of years after each impact. These long-lived hydrothermal systems provided stable environments where complex chemistry could occur, despite the chaos raging on the surface above. But the most intriguing evidence suggests that the bombardment may have been precisely calibrated to enable rather than prevent the emergence of life. The timing, intensity, and chemical composition of impactors appear optimized to deliver necessary materials while avoiding complete planetary sterilization. This apparent fine-tuning raises questions about whether the bombardment resulted from purely random processes or represents evidence of deliberate engineering on solar system scales. The first life. Somewhere between 4.1 and 3.5 billion years ago, in conditions hostile to modern life, chemistry became biology. This transformation remains the deepest mystery in planetary science. Despite decades of research, scientists have not identified the specific chemical pathways that led from simple organic molecules to self-replicating systems capable of evolution. The oldest widely accepted evidence for life comes from stromatolites, layered rock structures created by microbial mats that trapped and bound sediment particles. Modern stromatolites still form in a few locations worldwide, providing analogs for understanding their ancient counterparts. Fossil stromatolites from Western Australia date to 3.5 billion years ago, indicating that life had not only originated, but had already developed into complex microbial communities by that time. But evidence suggests life may be far older. Those controversial carbon isotope signatures from Greenland rocks indicate biological activity as early as 3.8 billion years ago. Even more provocatively, graphite inclusions in 4.1 billion-year-old zircons from Australia show carbon isotope ratios that some scientists interpret as biological in origin. If this oldest evidence proves valid, then life originated within 500 million years of Earth's formation, and possibly within just 100 million years of the planet developing liquid water oceans. This rapid emergence suggests either that life forms easily under suitable conditions or that the chemical precursors for life existed in the protoplanetary disk before Earth even finished forming. The specific environment where life originated remains unknown. Traditional theories favored shallow tidal pools, where wet-dry cycles could concentrate organic molecules and promote polymerization reactions. But discovery of thriving microbial ecosystems around deep ocean hydrothermal vents has shifted scientific focus toward these extreme environments. Hydrothermal vents provide several advantages as sites for life's origin. They offer abundant chemical energy from the reaction between hot mineral-rich vent fluids and cold seawater. They create steep thermal gradients that can drive chemical reactions and provide opportunities for thermophoresis, the concentration of molecules and temperature gradients. They exist independently of sunlight, allowing life to originate even when the atmosphere was opaque to solar radiation. The minerals forming at hydrothermal vents also provide templates and catalysts for organic chemistry. Iron-sulfur minerals common at vents can facilitate the synthesis of organic molecules and may have served as primitive metabolic catalysts before biological enzymes evolved. Some scientists propose that the first cells were actually mineral compartments within vent chimneys where organic chemistry was concentrated and protected. Alternative theories suggest life originated in shallow volcanic pools on land where wet-dry cycles provided opportunities for concentrating and polymerizing organic molecules. These environments offer ultraviolet radiation that can drive chemical reactions and opportunities for creating lipid membranes that could serve as primitive cell boundaries. Regardless of where life originated, the first organisms were undoubtedly simple. Early life probably consisted of RNA molecules that could both store genetic information and catalyze chemical reactions, solving the chicken and egg problem of which came first, genes or enzymes. This RNA world eventually gave way to the modern system where DNA stores information, RNA transmits it, and proteins perform most catalytic functions. The genetic code used by all life on Earth appears to have been established very early and has remained remarkably stable for billions of years. This universal code suggests that all life descended from a single common ancestor, often called LUCA, the last universal common ancestor. Luca lived approximately 3.5 to 4 billion years ago and possessed sophisticated cellular machinery, including ribosomes, membrane pumps, and metabolic pathways. But here's where the mystery deepens. The genetic and biochemical complexity displayed by Luca seems impossibly advanced for the earliest life. Luca possessed hundreds of genes and sophisticated molecular machinery that must have required hundreds of millions of years to evolve. This complexity suggests either that life originated much earlier than 4 billion years ago, or that evolution operated far more rapidly during Earth's earliest periods. Recent discoveries of ancient genetic sequences preserved in modern organisms reveal that early life developed sophisticated DNA repair mechanisms, radiation resistance, and stress response systems. These capabilities seem specifically designed for surviving in the harsh conditions of early Earth, suggesting that life faced extreme challenges from its earliest moments and evolved robust systems for ensuring survival. The rapid emergence and sophisticated nature of early life raises profound questions. Did life truly originate on Earth, or was it delivered from elsewhere in the solar system or beyond? The panspermia hypothesis proposes that life or its precursors traveled between worlds on meteorites, potentially seeding Earth with organisms or organic molecules that jump-started terrestrial biology. Evidence for panspermia includes the discovery of organic molecules and even amino acids in carbonaceous meteorites. Some meteorites contain water-altered minerals, indicating they came from bodies with liquid water, where biological processes could have operated. Hence, the survival of bacteria in space conditions for extended periods demonstrates that interplanetary transfer of living organisms is at least physically possible. The oxygenation catastrophe. For nearly 2 billion years after life's origin, Earth's atmosphere contained virtually no oxygen. Early life thrived in this oxygen-free world, extracting energy from chemical reactions involving hydrogen, methane, sulfur compounds, and iron. But approximately 2.4 billion years ago, everything changed. Oxygen began appearing in Earth's atmosphere in quantities that would prove deadly to most existing life forms. This transformation, called the Great Oxygenation Event, represents the most dramatic change in atmospheric chemistry in Earth's history. Oxygen levels rose from less than 0.001% to approximately 1 to 2% of the atmosphere within perhaps just a few million years. This change may seem modest compared to today's 21% oxygen atmosphere, but it represented a 1,000-fold increase that fundamentally altered the chemistry of the entire planet. The source of this oxygen was cyanobacteria, photosynthetic microorganisms that had evolved the ability to extract energy from sunlight by splitting water molecules. Photosynthesis releases oxygen as a waste product, and for hundreds of millions of years before the Great Oxygenation Event, cyanobacteria had been producing oxygen that was immediately consumed by reactions with dissolved iron and other reduced compounds in the oceans. But eventually, all the readily available reduced compounds became oxidized. Dissolved iron precipitated out of the oceans as iron oxide, creating the massive banded iron formations that now provide most of the world's iron ore. Once these chemical oxygen sinks became saturated, oxygen began accumulating in the atmosphere with dramatic consequences. For most existing life, oxygen was poison. This highly reactive gas destroyed biological molecules and disrupted cellular chemistry that had evolved in oxygen-free conditions. The Great Oxygenation Event triggered a mass extinction that eliminated most of Earth's microbial species, an apocalypse that scientists have called the oxygen catastrophe or oxygen holocaust. But this catastrophe also created opportunities. Oxygen enabled aerobic respiration, a metabolic process that extracts far more energy from organic molecules than anaerobic metabolism. Organisms that could tolerate or utilize oxygen gained enormous competitive advantages, driving evolution in entirely new directions. The oxygenation event also triggered dramatic climate changes. Methane, a powerful greenhouse gas that had helped keep early Earth warm despite the sun being 25% dimmer than today, reacted with oxygen to form carbon dioxide and water. The loss of atmospheric methane caused Earth's temperature to plummet, triggering the first global glaciation event. Evidence for this ancient ice age comes from glacial deposits found in rocks dating to 2.4 billion years ago. These deposits occur at tropical latitudes, indicating that ice extended from the poles to the equator in what scientists call a snowball Earth event. The planet may have remained frozen for millions of years before volcanic outgassing of carbon dioxide created enough greenhouse warming to melt the ice. The chemical signatures left by the Great Oxygenation Event are unmistakable. Banded iron formations abruptly cease forming as dissolved ocean iron became exhausted. Red beds, oxidized iron deposits, and terrestrial sediments appear for the first time. Sulfur isotope ratios in rocks show dramatic changes indicating that atmospheric chemistry had fundamentally transformed. But here's where the evidence becomes puzzling. The timing and magnitude of oxygenation seem remarkably precise, occurring exactly when Earth's chemical systems were prepared to handle the transformation without triggering runaway reactions that could have sterilized the planet. The process appears coordinated and controlled rather than chaotic and random. Some researchers have noted that the Great Oxygenation Event coincides suspiciously with the stabilization of continental crust and the beginning of modern plate tectonics. This correlation suggests that planetary geological processes and atmospheric chemistry may have been deliberately synchronized to create optimal conditions for the next phase of evolution. The oxygenation also enabled the development of the ozone layer, which shields the surface from harmful ultraviolet radiation. This protection allowed life to colonize shallow waters and eventually land surfaces that had previously been sterilized by UV radiation. The timing of these developments suggests a carefully orchestrated sequence of events rather than random chemical evolution. After the initial oxygenation event, oxygen levels remained relatively low for another billion years before rising again approximately 800 million years ago in a second oxygenation event. This pattern of staged increases suggests a regulated process rather than simple biological productivity driving atmospheric changes. The Boring Billion. Between approximately 1.8 and 0.8 billion years ago, Earth entered a period of remarkable stability that scientists have nicknamed the Boring Billion. During this enormous span of time, atmospheric composition, ocean chemistry, and global temperature remained relatively constant. Evolution appeared to slow dramatically, with few major innovations in life forms or ecosystems. This stability seems paradoxical. Earth's geological and biological systems are complex and dynamic, normally exhibiting constant change across multiple timescales. Yet for a billion years, the planet maintained equilibrium conditions that varied far less than periods before or after this interval. The Boring Billion coincided with the assembly and persistence of the supercontinent Rodinia, a massive landmass that gathered most of Earth's continental crust into a single block. supercontinent formation affects planetary systems in multiple ways, altering ocean circulation patterns, atmospheric circulation, and the cycling of nutrients between continents and oceans. Geological evidence indicates that plate tectonic activity slowed considerably during the Boring Billion. Continental collision rates decreased, volcanic activity diminished, and mantle convection patterns stabilized. This reduced geological activity limited the recycling of nutrients and the outgassing of volcanic carbon dioxide that normally drives climate variations. Ocean chemistry during this period shows unusual stability. Chemical proxies indicate that oxygen levels in deep ocean waters remained consistently low, creating persistent anoxic conditions that limited the ecological niches available for complex life. The oceans existed in a stable euxinic state, containing dissolved hydrogen sulfide in deep waters, while surface waters contained moderate oxygen levels. Life during the Boring Billion consisted almost exclusively of single-celled organisms and simple multicellular forms. Despite having had nearly 2 billion years to evolve since life's origin, organisms showed relatively little morphological complexity. The most advanced life forms were probably simple filaments and mats of photosynthetic bacteria and early eukaryotic cells. But beneath this surface stability, crucial evolutionary innovations were occurring. Eukaryotic cells, featuring complex internal structures and the ability to engulf and incorporate other cells, evolved during this period. The process of endosymbiosis, where one cell permanently incorporated another as an internal organelle, created mitochondria and chloroplasts that would later power complex multicellular life. Sexual reproduction evolved during the Boring Billion, allowing organisms to recombine genetic material and dramatically increasing the pace of evolutionary innovation. The origin of sex represents one of the most important evolutionary transitions, yet it occurred during a period of apparent evolutionary stasis. The stability of the Boring Billion may actually have been necessary for these complex innovations to develop. Rapid environmental change selects for short-term adaptations and survival strategies. The stable conditions during this period allowed evolution to experiment with complex cellular machinery and reproductive strategies that would have been eliminated by natural selection in more variable environments. Chemical evidence suggests that the Boring Billion was not entirely stable. Isotope ratios in rocks show variations indicating periodic perturbations to ocean chemistry and atmospheric composition. These variations may represent attempted evolutionary innovations that failed to establish permanent changes to planetary systems. Some researchers have proposed that the Boring Billion represents a period when biological and geological processes achieved perfect balance, creating a homeostatic planetary system that resisted change. This balance was eventually disrupted by the breakup of Rodinia and the resulting geological upheaval that triggered the next great transformation of Earth's environment. But here's where the pattern becomes suspicious. The Boring Billion appears precisely calibrated to last long enough for complex cells to evolve, while avoiding conditions that would have either sterilized the planet or locked it into permanent stasis. The duration and stability of this period seem designed to serve as an evolutionary incubation chamber. The end of the Boring Billion came suddenly. Approximately 800 million years ago, oxygen levels began rising again, continents began fragmenting, and the climate destabilized dramatically. These changes set the stage for the emergence of complex multicellular life and the beginning of Earth's modern biological era. Snowball Earth, between 720 and 635 million years ago, Earth experienced the most extreme ice age in planetary history. During this period, ice extended from the poles to the equator, covering the entire planet in glaciers, sometimes exceeding 1 kilometer thick. Oceans froze solid to depths of several hundred meters. Global average temperatures dropped to -50 degrees Celsius. Earth had become a snowball. Evidence for these global glaciations comes from glacial deposits found at tropical latitudes in rocks dating to this period. These deposits contain distinctive features including dropstones, large rocks carried by icebergs and dropped into fine marine sediments, and diamictites, unsorted sedimentary rocks created by glacial processes. The global distribution of these deposits indicates that ice reached the equator. Chemical evidence supports complete glaciation, carbon isotope ratios in rocks from this period show dramatic excursions indicating the near-complete shutdown of the biological carbon cycle. When ice covered the oceans, photosynthesis essentially ceased, allowing carbon isotope ratios to drift to values impossible in a biologically active world. The trigger for these snowball events appears to have been the breakup of the supercontinent Rodinia. Continental rifting and volcanic activity released enormous quantities of fresh rock that underwent chemical weathering, consuming atmospheric carbon dioxide and weakening Earth's greenhouse effect. As temperatures dropped and ice expanded from the poles, the highly reflective ice surface bounced sunlight back into space, accelerating cooling in a runaway feedback process. Computer models indicate that once ice extended past approximately 30 degrees latitude, the ice albedo feedback became irreversible. The planet rapidly froze completely and should have remained frozen indefinitely. The sun was still about 6% dimmer than today, providing insufficient energy to melt global ice coverage even with maximum greenhouse gas concentrations. Yet, the ice eventually melted. The solution to this paradox lies in volcanism. Even with frozen oceans, volcanic activity continued, steadily releasing carbon dioxide into the atmosphere. With photosynthesis shut down and chemical weathering halted by ice coverage, volcanic carbon dioxide accumulated without removal, eventually reaching concentrations perhaps 350 times higher than today. This massive greenhouse effect finally overcame the ice albedo feedback, triggering rapid deglaciation. Models suggest that temperatures could have swung from -50 to +50 degrees Celsius within just a few centuries as the ice melted. Glacial deposits are directly overlain by carbonate rocks that formed when the extreme atmospheric carbon dioxide reacted with rocks in a super greenhouse world. The snowball Earth events should have sterilized the planet, and most photosynthetic organisms would have been eliminated when ice cut off sunlight. Ocean freezing should have destroyed marine ecosystems. The temperature extremes during glaciation and deglaciation exceeded the tolerance ranges of most known organisms. Yet life survived. Microbial communities must have persisted in refugia, protected environments where liquid water and energy sources remained available. Possible refugia include deep ocean hydrothermal vents, volcanic hot springs that remained active beneath the ice, and thin bands of open water near the equator where volcanic heat prevented complete freezing. The most intriguing evidence suggests that life not only survived, but actually diversified during the snowball events. Genetic analysis indicates that crucial evolutionary innovations, including the development of multicellular animals, occurred during or immediately after these glaciation periods. And the extreme environmental stress may have accelerated evolution by creating intense selection pressures. But here's where the timing becomes suspicious. The snowball Earth events occurred precisely when conditions were optimal for triggering the evolution of complex, multicellular life. The environmental stress was severe enough to drive rapid evolution, but not so extreme as to cause complete extinction. The duration was long enough to force major evolutionary innovations, but short enough to avoid permanent planetary sterilization. Some researchers have noted that the chemical conditions following the snowball events were ideal for supporting large, complex organisms. Ocean chemistry changed dramatically, with increased oxygen and nutrient availability creating opportunities for evolutionary experimentation. The timing suggests that the snowball events may have been engineered to trigger specific evolutionary outcomes. The escape from snowball conditions also appears precisely calibrated. Volcanic carbon dioxide accumulated at exactly the rate needed to eventually trigger deglaciation without causing runaway greenhouse warming that could have boiled away the oceans. The feedback mechanisms that controlled this process show sophistication that seems unlikely to result from random planetary chemistry. The Cambrian explosion. Approximately 541 million years ago, life on Earth underwent the most dramatic transformation in its history. Within a geological instant of perhaps 10 to 25 million years, virtually all major animal body plans appeared in the fossil record. This event, called the Cambrian explosion, saw the sudden emergence of complex organisms with shells, skeletons, eyes, limbs, and sophisticated nervous systems. Before the Cambrian, life consisted primarily of simple, soft-bodied organisms, including microbial mats, algae, and the strange Ediacaran fauna that resembled nothing alive today. The Ediacaran organisms were mostly flat, pancake-shaped creatures that lived on the ocean floor, possibly absorbing nutrients directly through their body surfaces. They showed no obvious relationship to modern animal groups. Then, suddenly, the fossil record explodes with complexity. Trilobites appear with segmented bodies, compound eyes, and jointed appendages. Anomalocaris, a meter-long predator with grasping appendages and sophisticated vision, dominated early Cambrian seas. Creatures with shells, internal skeletons, and diverse feeding strategies filled ecological niches that had remained empty for billions of years. The Cambrian explosion is so dramatic that Darwin himself recognized it as a problem for his theory of evolution. Natural selection should produce gradual change, with organisms slowly developing new features over millions of years. Yet the Cambrian sees complex innovations appearing almost simultaneously across multiple unrelated lineages. Traditional explanations focus on ecological and environmental triggers. Rising oxygen levels may have crossed a threshold allowing large, active animals to evolve. The evolution of predation created an evolutionary arms race, driving rapid development of defensive structures like shells and protective spines. Genetic innovations, including Hox genes that control body plan development, may have unlocked evolutionary potential that had been previously constrained. But these explanations struggle to account for the scale and precision of the Cambrian diversification. The emergence of complex eyes in multiple independent lineages within a few million years seems impossibly rapid. The sudden appearance of mineralized skeletons and organisms that had existed for hundreds of millions of years without them suggests environmental triggers rather than gradual evolutionary processes. Chemical evidence indicates that ocean chemistry changed dramatically just before the Cambrian explosion. Phosphate concentrations increased, providing materials for skeleton construction. Calcium levels rose, enabling the formation of carbonate shells. Oxygen penetrated into deeper ocean waters, eliminating anoxic zones that had limited ecological opportunities. The timing of these chemical changes correlates precisely with the biological innovations of the Cambrian explosion. This correlation suggests that environmental engineering prepared the oceans for supporting complex animal life. The changes appear coordinated and systematic rather than resulting from random geological processes. Genetic analysis reveals another mystery. The genes controlling animal body plan development were already present in organisms that existed before the Cambrian explosion. These creatures possessed the genetic toolkit for building complex bodies, but didn't express these capabilities until the Cambrian. Something activated latent genetic potential that had existed unused for millions of years. The fossil record shows that Cambrian organisms appear fully formed in the oldest rocks containing their remains. There are no obvious transitional fossils showing gradual development of complex features. Eyes, for example, appear in the fossil record already sophisticated and functional, not as simple light-sensitive patches that gradually refined into complex optical systems. But here's where the evidence becomes most puzzling. The Cambrian explosion occurred during a period of unusual environmental stability. Climate was moderate, sea level was high, and ocean chemistry was favorable. Yet previous periods with similar conditions saw no comparable evolutionary innovation. Something specific about the Cambrian triggered explosive diversification that cannot be explained by environmental conditions alone. Some researchers have noted that the body plans established during the Cambrian explosion have remained remarkably stable for 540 million years. Virtually all subsequent animal evolution represents variations on themes established during this brief period. No fundamentally new body plans have evolved since the Cambrian, suggesting that this event established permanent constraints on animal architecture. The precision and coordination of the Cambrian explosion suggests the operation of processes beyond conventional evolution. The simultaneous appearance of complex innovations across multiple lineages, the activation of previously latent genetic capabilities, and the permanent establishment of body plan diversity all point toward directed, rather than random processes. The colonization of land. For the first 4 billion years of Earth's history, life remained confined to water. The land surface was barren rock, sand, and volcanic ash, swept by winds and sculpted by rain, but devoid of any living organisms. Then, approximately 470 million years ago, during the Ordovician period, life began its conquest of the continents. The first terrestrial colonizers were likely cyanobacteria and other photosynthetic microorganisms that could tolerate periodic drying and intense ultraviolet radiation. These pioneering organisms formed thin biological crusts on rocks and soil, beginning the slow process of transforming lifeless mineral surfaces into living ecosystems. Plants followed the microorganisms onto land. The earliest land plants were simple organisms, resembling modern liverworts and mosses, lacking true roots, stems, and leaves. These plants required constant moisture and could only survive in wet environments near water bodies. But they represented a crucial step in terrestrial colonization. By 420 million years ago, more sophisticated plants had evolved. Cooksonia, one of the earliest vascular plants, possessed simple branching stems containing specialized tissues for transporting water and nutrients. This innovation allowed plants to grow taller and colonize drier environments away from water sources. Within 50 million years, forests of primitive plants covered the continents. The colonization of land by plants triggered dramatic changes in Earth's geological and atmospheric systems. Plant roots broke down rock, dramatically accelerating chemical weathering and the formation of soil. Photosynthesis on land added to oceanic oxygen production, driving atmospheric oxygen levels to unprecedented heights. But the transformation went beyond simple chemistry. The burial of plant material in swamps created vast cold deposits that locked carbon away from the atmosphere. This carbon burial reduced greenhouse gas concentrations, triggering climate cooling that led to ice age conditions. The weathering of rock by plant roots consumed additional carbon dioxide, accelerating the cooling trend. Animals followed plants onto land. The first terrestrial animals were arthropods, including primitive relatives of spiders, mites, and centipedes. These creatures fed on decaying plant material and each other, establishing the first terrestrial food webs. By 360 million years ago, the first vertebrates, primitive amphibians, descended from lobe-finned fish, had made the transition from water to land. The evolution of animals capable of living on land required numerous physiological innovations. Terrestrial organisms needed mechanisms for preventing water loss, supporting their bodies against gravity without buoyancy from water, breathing air rather than extracting oxygen from water, and reproducing without water for fertilization and development. The fossil record shows that these innovations evolved relatively quickly once the colonization of land began. Within approximately 100 million years, terrestrial ecosystems achieved complexity rivaling marine systems, with diverse plants, herbivores, predators, and decomposers, forming intricate ecological networks. The Devonian period, 419 to 359 million years ago, saw the development of the first forests. Trees, including Archaeopteris, reached heights exceeding 30 meters, creating forest canopies that shaded the ground and created new ecological niches. These forests dramatically altered the global carbon cycle, atmospheric composition, and climate systems. By the Carboniferous period, 359 to 299 million years ago, vast swamp forests covered equatorial regions. These forests produced the coal deposits that would later fuel human industrial civilization. The forests also drove atmospheric oxygen to levels perhaps as high as 35% compared to today's 21%, in enabling the evolution of giant insects, including dragonflies with 70-centimeter wingspans. But here's where the pattern becomes suspicious. The colonization of land occurred with remarkable efficiency and speed. Within just 100 million years, life progressed from simple microbial crusts to complex forest ecosystems with sophisticated food webs. This rapid transformation suggests either that evolution operates far more quickly than standard models predict, or that the process was guided by factors beyond random mutation and natural selection. The timing of land colonization also correlates precisely with rising oxygen levels and the development of the ozone layer that protected the surface from ultraviolet radiation. These changes appear coordinated to create optimal conditions for terrestrial life. The sequence of innovations needed for land colonization appeared in precisely the correct order with precise timing, saying the Great Dying. 252 million years ago, at the boundary between the Permian and Triassic periods, Earth experienced the most catastrophic mass extinction in the history of complex life. This event, known as the Permian-Triassic extinction, or simply the Great Dying, eliminated approximately 96% of all marine species and 70% of terrestrial vertebrate species. Life came closer to complete annihilation than at any other time in Earth's history. The extinction occurred with stunning rapidity. Geological evidence suggests that the main pulse of extinction lasted less than 60,000 years, and possibly as little as 20,000 years. This represents an instant in geological time, a catastrophic collapse of planetary ecosystems that transformed Earth into a near-lifeless wasteland. The trigger for this extinction appears to have been the eruption of the Siberian Traps, one of the largest volcanic events in Earth's history. Over perhaps 1 million years, volcanic eruptions in Siberia released approximately 4 million cubic kilometers of lava, covering an area larger than Europe with basalt up to several kilometers thick. But the lava itself was not the primary killer. The eruptions released massive quantities of carbon dioxide, sulfur dioxide, and other volcanic gases into the atmosphere. Carbon dioxide concentrations may have reached 2,000 parts per million, compared to pre-industrial levels of 280 parts per million. Global temperatures increased by 8 to 10 degrees Celsius, creating a super greenhouse world. The warming triggered multiple cascading environmental catastrophes. Ocean temperatures rose to levels that eliminated dissolved oxygen from deep waters, creating vast anoxic zones where only specialized bacteria could survive. Hydrogen sulfide produced by these bacteria poisoned the oceans and may have reached the atmosphere in toxic concentrations. The warming also destabilized methane hydrates, frozen methane deposits in ocean sediments. Methane release created additional greenhouse warming in a runaway feedback loop. Carbon isotope excursions in rocks from this period indicate massive releases of isotopically light carbon, consistent with methane hydrate destabilization. Acid rain from volcanic sulfur compounds acidified both terrestrial and marine environments. Ocean pH dropped dramatically, dissolving the calcium carbonate shells of marine organisms and causing widespread ecosystem collapse. The fossil record shows that reef systems completely disappeared and took 10 million years to recover. On land, forests died and were replaced by simple plants, including fungi, that thrived on decaying vegetation. The fossil record shows a dramatic spike in fungal remains at the extinction boundary, evidence of ecosystems dominated by decomposition rather than photosynthesis. Animals that survived were mostly small, adaptable species that could survive on limited resources. The extinction showed disturbing selectivity. Some groups were nearly completely eliminated, while closely related groups with similar ecological roles survived relatively intact. This pattern suggests that the extinction mechanism targeted specific biological characteristics rather than affecting all organisms equally. But here's where the evidence becomes most troubling. The Permian-Triassic extinction shows signatures that seem designed to reset planetary ecosystems. The selectivity eliminated dominant groups while preserving rare lineages that would later diversify to fill empty ecological niches. The extinction appears to have cleared the way for specific evolutionary outcomes. Chemical signatures in rocks from the extinction boundary include unusual concentrations of rare elements and isotope ratios that don't match patterns expected from purely volcanic activity. Some formations contain microscopic spheres of metals and minerals that resemble particles produced by industrial process rather than natural volcanic eruptions. The timing of ecosystem recovery also appears suspicious. Life rebounded with remarkable speed considering the severity of the extinction. Within just a few million years, complex ecosystems were re-established, and new groups, including the ancestors of dinosaurs and mammals, began their diversification. This recovery seems faster than would be expected from random evolutionary processes following such catastrophic disruption. The Great Dying cleared ecological niches occupied by Permian fauna and flora, allowing entirely new groups to dominate terrestrial and marine ecosystems. The The extinction appears to have been precisely calibrated to eliminate existing dominant groups while preserving genetic diversity necessary for subsequent evolutionary innovation.